Hello guys, welcome back to another video from NCERT Proberer. In this video, I am going to explain and discuss the chapter, Breathing and Exchange of Gases. As we all know, NCERT is essential for our main exams like board exams, NEET, KVPY exam etc. So in this video I will explain chapter, Breathing and Exchange of Gases line by line. Don't skip or close the video in between. Watch the full video carefully, it will increase your knowledge about this chapter. If you are new to my channel, welcome to my channel, NCERT Proberer. Subscribe my channel for more videos and press the bell icon for notification. So let's start. Chapter 17. Breathing and Exchange of Gases. As you have read earlier, oxygen is utilized by the organisms to indirectly break down nutrient molecules like glucose and to derive energy for performing various activities. Carbon dioxide, which is harmful, is also released during the above catabolic reactions. It is, therefore, evident that O2 has to be continuously provided to the cells, and CO2 produced by the cells have to be released out. This process of exchange of O2 from the atmosphere with CO2 produced by the cells is called breathing, commonly known as respiration. In most of the plants and animal species, the process of respiration at cellular level involves use of oxygen for oxidation and simultaneous release of carbon dioxide. This type of respiration is known as aerobic respiration. Such respiration may be direct, that is, exchange of gases takes place without any respiratory organs, or indirect, which involves respiratory organs such as skin, gills, lungs, etc. Place your hands on your chest, you can feel the chest moving up and down. You know that it is due to breathing. How do we breathe? The respiratory organs and the mechanism of breathing are described in the following sections of this chapter. Respiratory organs. Mechanisms of breathing vary among different groups of animals depending mainly on their habitats and levels of organization. Lower invertebrates like sponges, coelenterate, flatworms, etc., exchange O2 with CO2 by simple diffusion over their entire body surface. Sponges have a water transport system or canal system. Water enters through minute pores called ostia in the body wall into a central cavity spongicle. Earthworms use their moist cuticle, because the earthworm lacks specialized breathing devices. Therefore, in earthworms, respiratory exchange occurs through moist body surface into their bloodstream. Insects have a network of tubes called tracheal tubes to transport atmospheric air within the body. Insects are included in phylum Arthropoda. The respiratory organs of Arthropoda are gills, book lungs, or tracheal system. Special vascularized structures called gills are used by most of the aquatic arthropods and mollusks, whereas vascularized bags called lungs are used by the terrestrial forms for the exchange of gases. Among vertebrates, fishes use gills, whereas reptiles, birds, and mammals respire through lungs. Amphibians like frogs can respire through their moist skin. Amphibians also respire through gills and lungs. Mammals have a well-developed respiratory system. Human respiratory system. The human respiratory system consists of nose, pharynx, larynx, trachea, bronchi and lungs. Nose is a specialized organ at the entrance of the respiratory system, that is divided into an external portion and an internal portion. The external nose is the portion of the nose that visible on the face and consists of a supporting framework of bone, and hyaline cartilage covered with muscle and skin, and lined by mucous membrane. The interior structures of external nose have three functions: 1. Warming, moistening and filtering incoming air. 2. Detecting olfactory stimuli. 3. Modifying speech vibrations as they pass through the large, hollow resonating chambers. The internal nose is the portion of the nose consists of nostrils and nasal chamber. It leads to the nasal chamber through the nasal passage. The regions of nasal chamber are: A. Nasal septum that divides the nasal cavity into right and left sides. B. Nasal vestibule, which anterior portion of the nasal chamber. C. Nasal cavity, which superior part of the nasal cavity. D. Nasal conchae, 3 shelves formed by projections that extend out of each lateral wall of nasal cavity. Then the nasal chamber opens into nasopharynx, which is a superior portion of pharynx and common passage for food and air. Nasopharynx opens through glottis of larynx region into the trachea. Larynx is a cartilaginous box which helps in the sound production and hence called sound box. During swallowing, the pharynx and larynx rise. Elevation of the pharynx widens it to receive food or drink. Elevation of larynx causes the epiglottis to move down, and form a lid over the glottis, closing it off. When small particles of dust, smoke, food, or liquid pass into the larynx, a cough reflex occurs, usually expelling the material. Trachea or windpipe is a straight tube that is about 12 cm long and 2.5 cm in diameter, extending up to the mid-thoracic cavity, which divides at the level of 5th thoracic vertebra into a right and left primary bronchi. The layers of the tracheal walls from deep to superficial consist of mucosa, submucosa, hyaline cartilage, and adventitia. The primary bronchi divide to form smaller secondary bronchi. The secondary bronchi continue to branch, forming smaller tertiary bronchi that divides into bronchioles. The bronchioles contain Clara cells which may protect against harmful effects of inhaled toxins and carcinogens. Bronchioles in turn branch repeatedly, and the smaller ones branch into even smaller tubes called terminal bronchioles. The tracheae, primary, secondary and tertiary bronchi and initial bronchioles are supported by incomplete cartilaginous rings. Each terminal bronchioles gives rise to a number of very thin, irregular walled, and vascularized bag-like structures called alveoli. The branching network of bronchi, bronchioles and alveoli comprise the lungs. Each lung is enclosed and protected by a double-layered serous membrane called the pleural membrane. The outer layer, called parietal pleura, lines the wall of thoracic cavity. The inner layer, visceral pleura, covers the lungs themselves. Between the visceral and parietal pleurae is a small space, the pleural cavity, which contains a small amount of lubricating fluid secreted by the membranes. The functions of the pleural fluid is to reduce friction between the membranes, allowing them to slide easily over one another during breathing. The part starting with the external nostrils up to the terminal bronchioles constitute the conducting part, whereas the alveoli and their ducts form the respiratory, or exchange part of the respiratory system. The conducting part transports the atmospheric air to the alveoli, clears it from foreign particles, humidifies, and also brings the air to body temperature. Exchange part is the site of actual diffusion of O2 and CO2 between blood and atmospheric air. The lungs are situated in the thoracic chamber, which is anatomically an airtight chamber. The thoracic chamber is formed dorsally by the vertebral column, ventrally by the sternum, laterally by the ribs, and on the lower side by the dome-shaped diaphragm. The anatomical setup of lungs in thorax is such that any change in the volume of the thoracic cavity will be reflected in the lung or pulmonary cavity. Such an arrangement is essential for breathing, as we cannot directly alter the pulmonary volume. Respiration involves the following steps: 1. Breathing or pulmonary ventilation, by which atmospheric air is drawn in and CO2-rich alveolar air is released out. 2. Diffusion of gases O2 and CO2 across alveolar membrane. In this process, pulmonary capillary blood gains O2 and loses CO2. 3. Transport of gases by the blood. 4. Diffusion of O2 and CO2 between blood and tissues. In this step, the blood loses O2 and gains CO2. 5. Utilization of O2 by the cells for catabolic reactions and resultant release of CO2. Consume of O2 and give off of CO2 during production of ATP are termed as cellular respiration. Mechanism of breathing. Breathing involves 2 stages: 1. Inspiration, during which atmospheric air is drawn in. 2. Expiration, by which the alveolar air is released out. The movement of air into and out of the lungs is carried out by creating a pressure gradient between the lungs and the atmosphere. The breathing mechanism can be explained on the basis of Boyle's law, which gives the inverse relationship between volume and pressure. It states that the pressure of a gas in a closed container is inversely proportional to the volume of the container. This means that if the size of closed container is increased, the pressure of the gas inside the container decreases, and that if the size of the container is decreased, then the pressure inside it increases. Example: inspiration can occur if the pressure within the lungs is less than the atmospheric pressure, that is, there is a negative pressure in the lungs with respect to atmospheric pressure. Mammals undergo negative pressure breathing, that is, the lungs draw air due to reduction of pressure within them. This allows them to eat and breathe simultaneously. If air was to be forced into the lungs, it would carry food particles with it, causing the trachea to block. Expiration takes place when the interpulmonary pressure is higher than the atmospheric pressure. The diaphragm and a specialized set of muscles, external and internal intercostals between the ribs, help in the generation of such gradients. Inspiration is initiated by the contraction of diaphragm, which increases the volume of thoracic chamber in the anteroposterior axis. Contraction of diaphragm is responsible for about 75% of the air that enters the lungs during breathing. The contraction of external intercostal muscles lifts up the ribs and the sternum, causing an increase in the volume of the thoracic chamber in the dorso-ventral axis. Contraction of these muscles is responsible for about 25% of the air that enters the lungs during breathing. The overall increase in the thoracic volume causes a similar increase in the pulmonary volume. An increase in pulmonary volume decreases the intrapulmonary pressure to less than the atmospheric pressure, which forces the air from outside to move into the lungs. Relaxation of the diaphragm and the sternum to their normal positions and reduce the thoracic volume and thereby the pulmonary volume. This leads to an increase in intrapulmonary pressure to slightly above the atmospheric pressure, causing the expulsion of air from the lungs, that is, expiration. Normal expiration during breathing is a passive process because no muscular contractions are involved. It becomes active only during forceful breathing. On an average, a healthy human breathes 12 to 16 times per minute. The volume of air involved in breathing movements can be estimated by using a spirometer. Respiratory volumes and capacities. 1. Tidal volume. Volume of air inspired or expired during a normal respiration. It is approximately 500 milliliter. That is, a healthy man can inspire or expire approximately 6,000 to 8,000 milliliter of air per minute. 2. Inspiratory reserve volume. Additional volume of air, a person can inspire by a forcible inspiration. This averages 2,500 milliliter to 3,000 milliliter. 3. Expiratory reserve volume. Additional volume of air, a person can expire by a forcible expiration. This averages from 1000 milliliter to 1100 milliliter. 4. Residual volume. Volume of air remaining in the lungs even after a forcible expiration. This averages 1100 milliliter to 1200 milliliter. 5. Inspiratory capacity. Total volume of air a person can inspire after a normal expiration. This includes tidal volume and inspiratory reserve volume. 6. Expiratory capacity. Total volume of air a person can expire after a normal inspiration. This includes tidal volume and expiratory reserve volume. 7. Functional residual capacity. Volume of air that will remain in the lungs after a normal expiration. This includes expiratory reserve volume and residual capacity. 8. Vital capacity. The maximum volume of air a person can breathe in after a forced expiration. This includes expiratory reserve volume, tidal volume, and inspiratory reserve volume, or the maximum volume of air a person can breathe out after a forced inspiration. 9. Total lung capacity. Total volume of air accommodated in the lungs at the end of a forced inspiration. This includes residual volume, expiratory reserve volume, tidal volume, and inspiratory reserve volume or vital capacity and residual volume. Exchange of gases. Alveoli are the primary sites of exchange of gases. Exchange of gases also occur between blood and tissues. O2 and CO2 are exchanged in these sites by simple diffusion, mainly based on pressure or concentration gradient. Solubility of the gases as well as the thickness of the membranes involved in diffusion are also some important factors that can affect the rate of diffusion. A gradient is present for CO2 in the opposite direction, that is from tissues to blood and blood to alveoli. As the solubility of CO2 is 20 to 25 times higher than that of O2, the amount of CO2 that can diffuse through the diffusion membrane per unit difference in the partial pressure is much higher compared to that of O2. The diffusion membrane is made up of three major layers, namely, the thin squamous epithelium of alveoli, the endothelium of alveolar capillaries, and the basement substance in between them. All the factors in our body are favorable for diffusion of O2 from alveoli to tissues and that of CO2 from tissues to alveoli. Alveolus with pulmonary capillary. The walls of alveoli is very thin, and with the rich network of blood capillaries, it forms respiratory membrane or diffusion membrane. Three main layers constitute the diffusion membrane. They are squamous epithelium of the alveoli, the basement substance, and the endothelium of the pulmonary capillary. Transport of gases. Blood is the medium of transport for O2 and CO2. About 97% of O2 is transported by RBCs in the blood. The remaining 3% of O2 is carried in a dissolved state through the plasma. Nearly 20 to 25% of CO2 is transported by RBCs, whereas 70% of it is carried American Samoa bicarbonate. About 7% of CO2 is carried in a dissolved state through plasma. Transport of oxygen. Hemoglobin is a red-colored iron-containing pigment present in the RBCs. O2 can bind with hemoglobin in a reversible manner to form oxyhemoglobin. Each hemoglobin molecule can carry a maximum of 4 molecules of O2. Binding of oxygen with hemoglobin is primarily related to partial pressure of O2. Partial pressure of CO2, hydrogen ion concentration and temperature are the other factors which can interfere with this binding. A sigmoid curve is obtained when percentage saturation of hemoglobin with O2 is plotted against the PO2. This curve is called the oxygen dissociation curve and is highly useful in studying the effect of factors like PCO2, H and concentration, etc., on binding of O2 with hemoglobin. In the alveoli, where there is high PO2, low PCO2, lesser H and concentration and lower temperature, the factors are all favorable for the formation of oxyhemoglobin, whereas in the tissues, where low PO2, high PCO2, high H and concentration and higher temperature exist, the conditions are favorable for dissociation of oxygen from the oxyhemoglobin. This clearly indicates that O2 gets bound to hemoglobin in the lung surface and gets dissociated at the tissues. Every 100 ml of oxygenated blood can deliver around 5 ml of O2 to the tissues under normal physiological conditions. Transport of carbon dioxide. CO2 is carried by hemoglobin, as carbaminohemoglobin about 20 to 25%. This binding is related to the partial pressure of CO2. PO2 is a major factor which could affect this binding. When PCO2 is high and PO2 is low as in the tissues, more binding of carbon dioxide occurs, whereas, when the PCO2 is low and PO2 is high as in the alveoli, dissociation of CO2 from carbaminohemoglobin takes place, that is CO2 which is bound to hemoglobin from the tissues is delivered at the alveoli. RBCs contain a very high concentration of the enzyme, carbonic anhydrase, and minute quantities of the same is present in the plasma too. At the tissue site where partial pressure of CO2 is high due to catabolism, CO2 diffuses into blood, RBCs and plasma, and forms HCO3- and H+. At the alveolar site where PCO2 is low, the reaction proceeds in the opposite direction, leading to the formation of CO2 and H2O. Thus, CO2 trapped as bicarbonate at the tissue level and transported to the alveoli is released out as CO2. Every 100 ml of deoxygenated blood delivers approximately 4 ml of CO2 to the alveoli. Regulation of respiration. Human beings have a significant ability to maintain and moderate the respiratory rhythm to suit the demands of the body tissues. This is done by the neural system. A specialized center present in the medulla region of the brain, called respiratory rhythm center, is primarily responsible for this regulation. Another center present in the pons region of the brain, called pneumotaxic center, can moderate the functions of the respiratory rhythm center. Neural signal from this center can reduce the duration of inspiration, and thereby alter the respiratory rate. A chemosensitive area is situated adjacent to the rhythm center, which is highly sensitive to CO2 and hydrogen ions. Increase in these substances can activate the center, which in turn can signal the rhythm center to make necessary adjustments in the respiratory process, by which these substances can be eliminated. Receptors associated with aortic arch and carotid artery also can recognize changes in CO2 and H and concentration, and send necessary signals to the rhythm center for remedial actions. The role of oxygen in the regulation of respiratory rhythm is quite insignificant. Next is our last topic in this section. Disorders of respiratory system. Respiratory diseases, or lung diseases, are pathological conditions affecting the organs and tissues that make gas exchange difficult in air-breathing animals. They include conditions of the respiratory tract including the trachea, bronchi, bronchioles, alveoli, pleurae, pleural cavity, and the nerves and muscles of respiration. Respiratory diseases range from mild and self-limiting, such as the common cold, to life-threatening diseases. 1. Asthma. Asthma is difficulty in breathing causing wheezing due to inflammation of bronchi and bronchioles. Wheezing means breathing with a whistling or rattling sound in the chest. 2. Emphysema. Emphysema is chronic disorder in which alveolar walls are damaged, producing abnormally large spaces that remain filled with air during exhalation, due to which respiratory surface is decreased. Major causes of this is cigarette smoking, air pollution, and occupational exposure to industrial dust. 3. Occupational respiratory disorder. Damage to the lungs caused by dust or fumes of noxious substances inhaled by the workers in certain specific occupation. Long exposure can give rise to inflammation leading to fibrosis. Fibrosis means proliferation of fibrous tissues. Example for occupational respiratory disorders are as follows. 1. Asbestos-related diseases. Serious lung disorders that develop as a result inspiring asbestos particles. 2. Black lung diseases. Lungs appear black due to inspiration of coal dust over a period of many years. If you find this video helpful, like, share, and subscribe my channel for more videos, and expecting your reviews in comment section.